Charlotte Sirot, and Joachim and I are very sorry we were not able to be here with you today, but we wanted to share with you some of our main results of the METRODIVER project, which was interested to investigate the services of marine protected areas through the lens of trophic diversity. So in this project, we investigated this question through three ecosystem level. So at community level, population level, and digestive microbiota level. And each of this package or community were led by three European labs. So the CRIOB at Perpignan in France for the community, the GMIT in Ireland for the fish population, and the ULB for the digestive microbiota in Belgium. And we, despite the fact that we had three different levels, we tried to gather all the information we got from these different ecosystem levels, and we tried to correlate these data to different services that we can find, knowing like productivity, spillover, nutritional quality for fish flesh, nutrient recycling, and individual health. So as I told you, three different work package, and for this presentation I will focus more on work package 1 for clarity reasons. It is important to remind you that the METRODIVER were sampled in three different areas in France with MPAs. So the first was the Bonifacio MPAs, the Blue Coast near Marseille, and the third one is focused on the Vermeil Coast region around Banyuls. In this region we can see two main MPAs, so Banyuls, as I told you, and the Cap de Creus. And both of them have fully protected area as well as partially protected areas. For work package 1, there was 23 UVC sites. So UVC means underwater visual sensors that provides divers to dive and count the different fish from the different species, and also to measure them to estimate the biomass. For work package 2 and 3, 15 fishing zones have been investigated with local fishermen. So as I told you, I will focus on work package 1 with the first objective that have been to set up a new statistical method to characterize the spillover. And we choose the INLA model, that is a Bayesian model that can take in account the autocorrelation of spatial data. And it was the first time for characterizing the spillover that this method have been used. And I wanted a bit to make you know how it works. Just a quick explanation. Basically, it's kind of a mixed model with actually an autocorrelation through this mesh. So just to show you, here is the coast and here is the mesh that's allowed to take into account the autocorrelation of spatial data. And once we have this INLA method, we decided to characterize the spillover, and first the qualitative spillover. So here you have the spillover model on biomass, and you can see that the coefficient that correspond to the distance to Banyuls MPA centroid is significantly different than 0. So you have here a significant effect of the distance to explain the biomass. And the novelty here is that we also explain the nutrient spillover, so a qualitative spillover, and that we tried to know whether some nutrients were more exported or less exported than others. And here you can see the Bayesian model, the INLA model for the omega-3. And here you can see that the zone has an effect on the quantity of omega-3, meaning that for the omega-3, the export of this nutrient is not only related to biomass. So the conclusion of what I showed you here was that MPA not only export biomass but also provide specific nutrient to local fisheries, and that's what I explained with the omega-3 nutrient. This will be the focus on the paper 2, and we are now gathering all the teams to understand this conclusion in light of the trophic diversity or processes change. We also wanted to see whether there was a change in fish prey, and here we are collaborating with Work Package 2. And we also wanted to see whether fish microbiota could explain this modification. So with Work Packet 3, that already published 2 papers that you can find in the literature now. Well, if you have other questions, because it's very fast, it's very quick, don't hesitate to send me a message, an email, and I thank you very much for your attention. Bye.